I'm Axel Hausmann from the Natural History Museum in Munich, another lepidopterist, and my research focus is on geometric moths, DNA barcoding, and in the last time a bit more also entering applications in meta-barcoding. You can see here the list of numbers. I don't want to read everything, just highlight 190,000 sequences with 23,000 bins. I will come back to that later. The German barcode network basically mainly is based on the two large projects in Bavaria and in Germany, DFB and iBOL. The lead is in Munich and Bonn, involving about 50 researchers, many associates, 16 institutions. We have got about 13 million euro for these projects in Germany. And of course, we have other barcoding projects in foreign countries. There is one in Indonesia, and and then there is another project that started this year. I will come back to that later, taxonomics. They have got 10 million euro of funding, but not everything goes to DNA barcoding. I will come back to that later. There are infrastructure projects. Senckenberg Museum is building up a new facility on genomics. We still don't know which kind of projects will be running there, but we hope that also this will involve DNA barcoding. So the future prospects, we still hope that to get a third period for iBOL. Now, with the insect decline hype, we hope to have a possibility there. The museum in Bonn already has got 2 grants for monitoring. We have a lot of, several meta-barcoding applications projects running. There are smaller projects in Bavaria. And in the end, I will present you a plan for doing type barcoding in German museums. So that's for the future. First project, the project in Bavaria started in 2009, hosted in our museum in Munich with 7 curators involved. We have got 1.5 million euro plus our salaries. After 2019, we hope to continue that with institutional funds and money from associations and smaller grants for monitoring to complete the barcode library. Oh no, that was not finished. The target is all animals, and we have done 20,000 species, which is 57% of the Bavarian fauna in 100,000 specimens that have been barcoded. The second project, 3 years later, came for the whole territory of Germany. The lead is in Bonn and Munich. It involves about 50 curators and scientists, 16 institutions, altogether funded by the German Ministry of Science with 11 million euro. And the outcome you see here, out of the 48,000 German species, and the target was to make close to 200,000 barcodes. We have done 250,000 barcodes, and the coverage is currently at 50%, 24,000 species of the German fauna barcoded. This is sufficient to do a lot of applications which were in the focus of the second phase of iBOL. To break it down to smaller groups, to orders, you see that the large insect orders are well represented with 5,700 bins in the Hymenoptera, in the Diptera, with a lot of dark taxa involved. Hymenopterans also well represented with wasp and bees complete. Lepidopterans almost complete, and the beetles with 75% also well represented. The second, another project is on the geometrics of the world. I'm doing this together with 150 members of Forum Herbulo initiative, and we have assembled 23,000 bins for geometric moths. I think it's top scoring on BOL, no? Isn't it?
Maybe sessiomidas are threatening you.
So, if you compare it with the number of described species, which is also 23,000, this makes clear that this is an excellent basis for taxonomy and also makes the geometrid moths as a wonderful model group for biodiversity research and for ecological research. This is another project still in the phase of application. DCOL, digitizing German nature history collections. We hope to use that for DNA barcoding of type specimens. We hope to get about 5 million euro, for perhaps more, to barcode these type specimens. We have already done 4,000, and we want to add 10,000 primary type specimens from our museum, and still we hope to involve other museums from Germany. Altogether we will have about 30,000 primary type specimens and 200,000 paratypes. Let's see how much money they will give us and do that. And the last project I want to mention is the project Taxonomics, with 27 single projects mainly done by postdocs and PhD. This is from the DFG, the German Association for Funding projects, with 5.5 million euro, but only a part of that goes to DNA barcoding. The aim of this project is to improve the interface between taxonomy and genomics. And there are several projects aiming at improving the standards for NGS sequencing of old collection material. Coming to the SWOT analysis, you may be surprised to read here the federal structure of Germany as a strength, but you have to consider that the first project was on Bavaria. And the northern Germans don't like to see Bavaria on the top. So they extended the project to the whole of Germany. And then the Bavarians said that we have also to extend our project. And this was a ping-pong game to the benefit of DNA barcoding in Germany. Why not? And the strength, of course, is the high coverage of national fauna, with 50% of the species done, which is sufficient to do, as I already said, to do meta-barcoding applications, and several stakeholders already do that in environmental monitoring, in pest control, in forensics, in food security, and others. Of course, we have many large nature history collections and many experts in Germany, which is also a strength. And the last strength I want to add is partnership and friendship with you. So, weaknesses. There is little coordination outside the iBOL project currently, which can be improved. We have poorly represented the soil organisms, the mites, the marine fauna, fungi, and protists, but that may be similar in some other countries. And there is a poor taxonomic identification and calibration in dipterans and parts of the hymenopterans. Threats. The large projects on Bavaria and Germany will be finished in 2019, though we still hope to have the iBOL project extended for another 3 years. The legal issues Nagoya Protocol already said, which is a huge problem to help other countries. For us. The leading directors in Munich and Bonn will retire next year and in 4 years. Also a threat. The opportunities are that we have a strong recommendation of the National Academy of Sciences of using DNA barcodes for biological research, and the actual hype around the insect biomass decline will help us, and I'm very optimistic to raise other foods. The economy of Germany is not that bad. Technology and applications are well accepted among scientists and stakeholders. The museum curators are no more reluctant of giving us tissues of specimens, type specimens. And last but not least, we have several established international cooperations that may help in future to do other projects. So thank you very much.